afternoon, everyone, to our last fourth talk of the semester. I hope you've been enjoying these talks during the whole year, really. We'll resume the seminars next semester, probably after the summer. But in the meantime, just to close this series of seminars this year, I'm really, really happy that we have Ada Cavalcanti today here to give us the talk. So Ada got her PhD in Italy, and then went for a master's at the University of Pennsylvania in the US, then took a PhD in biology in 2005 in Heidelberg University. She then moved to the Max Planck Institute, and since 2011, she is a group leader at the Max Planck as well. She's also a part of the Faculty of Biosciences at the Heidelberg University, and she's done very, very beautiful work on the mechanobiology of integrins and receptors and growth factors, and essentially how cells communicate with the extracellular matrix. So it's very, very, very beautiful work, which I'm sure she's going to give us a taste of that today. So thank you, Ada, for accepting to give a talk here. And as usually, when we're going to have a series, 10 minutes or whatever it's allowed for time for questions at the end. But in the meantime, Ada, the screen is yours. Thank you very much.
So thank you, Sergi, for a really kind invitation. It's a pleasure to be here today, even if it's virtual. I'm still very happy, and I'm really looking forward to having some nice discussion immediately after the talk, or if some questions might pop up in your mind, please don't hesitate to drop me a line. Also happy to have more Zoom meetings now that we are in a Zoom here, and talk to students about receptors and anything that is related to receptor forces. So I start sharing the screen. If you have questions, feel free to interrupt me anytime. Otherwise, we talk at the very end. So what I will tell you today, as Sergi said, is of course what is in the heart of our research. It's a little bit how we developed actually some ways to look at receptor-mediated forces, and in particularly with emphasis on like local control of these mechanical interactions. So first of all, it's like why we look actually at cells at the end and their environment and what is surrounding cells. And this is really a complex picture, because there are not only matrix proteins, also neighbors cells, a lot of concomitant events that take place where both physical signals that are mediated by matrix proteins, but also soluble signals that come along with embedded factors such as growth factors, cytokines, and chemokines. They have to converge somehow to a cell, trigger signals and transcription events or expression of genes. But then in the end, they regulate a bunch of, or many cellular functions, such as those that are listed here. And in the end, this gets back integrated to a larger scale, for example, for tissue formation, maintenance of tissue, homeostasis, or regeneration. And the central role in this integration of signals, to answer this question, actually, how also the adaptation to these changes in the environment, the central role is carried out by cell surface receptors. The major classes of receptors that we'll know involved in adhesion are integrins for the matrix-mediated adhesions, and then at adherence junction receptors such as ecoadherins. And the common trait of these receptors, so keep in mind, is that they share a lot of similarities in terms of mechanics, saying that they do not really function alone. They cluster and recruit a lot of proteins at the intracellular level and link the actin cytoskeleton. And in the end, our ultimate goal is to see how these two compartments are coordinated, but we are not really that far in looking at this at a molecular detail we would like to have. But we're still stuck with the first question that I posed here. My talk will be basically really centered around how, in the end, we look at the integration of different signals, first from the ECM, and then in the second part of my talk, between neighboring cells, keeping really focus on the regulation of forces in cells. So for that, what we did basically when I started really working on this local control of receptors was to design some surfaces and materials that will allow us to look at this event and also to regulate the crosstalk of different types of receptors. And this is really like a summary slide of many types of materials. I call them bio-inspired materials. You may see already and appreciate that they're in the end a very artificial material, but they have somehow elements that we can nicely control and independently tune to then put, let's say, our findings into bigger pictures and extrapolate, for example, this information that we get from nano patterning of adhesive ligands or patterning of cells and controlling cell shape, and then integrating them with soft materials where we also control stiffness, for example, of the environment. At the same time, for the recording of forces, what we do use a lot in the lab, and I guess the audience is mainly familiar with traction force microscopy. We look at how cells really generate forces at the interface by monitoring displacement of fluorescent label beads, for example. And we also apply some molecular tension sensors that are inspired, by knowing the mechanics of certain protein domains, for example, or DNA strands. And we record the fluorescent signal here at the interface between cells and surfaces by just using some fret pairs, for example. But this is all extracellular. So if you know, a lot of beautiful work has been done, for example, by Carsten Grashoff, also with molecular tension sensor in intracellular level for vinculin or tallinn. But the principle is pretty much the same, that we have like two parts, and then as force is generated, the fluorophore gets away from a quencher, and you see this beautiful fluorescent signal that is due to force generation. So when I said like nanoscale control, the overall concept has been actually developed like with the nano pattern by Joachim Spatz, with whom I've been working for almost the last 20 years as I started working in Germany. And here the idea is pretty simple. So you take some nanoparticles that are produced by a technique called block copolymer micellar nanolithography. And the gold, through its very selective chemistry, serves as the anchor point for ligands, integrin ligands in this case. And in the end, one can create surfaces that have different interparticle distances and therefore regulate the lateral clustering of single integrins. I shall mention that these particles are as small as 8 to 10 nanometers, so only one receptor can fit in the binding site, but we might have multiple ligands, but in the end, just one receptor will get activated. And to force cells actually to recognize these binding sites, what we did, we applied a passivating layer. So these are really just single anchor points for receptors. And in this scanning electron microscopy, you can appreciate that really this is the points where, even if you think about electron microscopy preparation with some retraction, that it's inevitable, still these are the major anchor points to the surface. In these time lapses here, this is really super old stuff, but I like really to put some history to then understand what is like in the present and challenges that we still have to face in future work. The key message here is that as you increase the spacing between integrins, you basically still have cells that are adherent. As you see, this is like smaller spacing, intermediate and large. Intermediate is here and large here, where cells start sliding and adhesion looks very unstable. And this sliding is actually due to the fact that focal adhesions get highly dynamic. So this is a temporal coding with a ratio of the intensity of a focal adhesion protein, paxillin. And as we increase the spacing, you see, I call it this a Christmas tree effect, where you have really transient structures. And in trying to understand what makes these structures very transient, is it due, to having less number of bonds, where is really the failure, in keeping and growing stable focal adhesions? What we did, we observed over time the sliding speed, because we always observed that, this contact disassemble, but they kind of go in a centripetal direction, as shown here in these chymographs. And the sliding speed is kind of increasing as you increase the spacing. And now, knowing this, actually, we also have to say in this piece of work, if you're interested in all the theory behind, we also had to apply a Wells model at that time and try to reason that we need to have a critical fraction of bonds established at the adhesion sites to kind of harness the centripetal pulling from the actin, and probably due to retrograde flow. But you see, this is work done in 2010, and then it took us really a lot of years to start understanding what is the molecular regulation here. I have to say really upfront, there isn't really a magical protein that is missing and not coming there to stabilize this context, because we could track down the localization even to the last one that appear focal adhesions, such as 16, they will still show up in a larger spacing. But actually, we started suspecting that it's not really like molecular regulation, but rather like, as I mentioned to you, a force distribution at the bonds. And this force distribution actually is, again, these experiments are on glass. So if you see like contacts that are kind of, you take a snapshot, you see really nice elongated focal adhesions, what we are used to see. And then as we increase the space in the contacts, look really like dot-like in some cases, and cells shrink, as I showed you before in the previous movies. So now the thing is that, if you start thinking about forces, so one of the first steps is sensing the rigidity of the environment. And one way to really probe and see how cells sense the environment is to use materials that have different stiffness. So to combine what we had done, before with the patterning and now rigidity sensing, and trying really to understand the regulation of forces through integrins at the nanoscale, we did this little development. It was like to transfer our patterns from a glass to a soft material. The soft material is, in this case, a polyacrylamide that is widely used for traction force microscopy. And the idea is, in the end, to vary and have a soft and stiff environment, and then see how cells would spread and respond when we independently tune the spacing and the stiffness of the environment, while maintaining the same RGD adhesion type. And this was really puzzling because if you see here from the color code, with a 15 nanometer, or like even on a glass, so as you increase the stiffness on a rigid substrate, 50 is like the best, for the adhesion length. And what I showed you before, as you increase, you have like the shorter adhesions. However, if you start having soft materials and increase the spacing, you kind of shift the peak for the formation of adhesion length. So on soft materials and for larger spacing, we observed very long focal adhesions. From what I told you before, with all the stability, this was really like hard to understand for us when we did the experiments. We also monitor the cellular traction forces, which are shown here. And you see that even for larger spacing, cells are able to generate tractions when they are put on softer materials. And this is really, I think, the key in try to understand what is the linkage at the end between the cellular forces and the receptor cluster regulation. And in this case, modeling came really, it helped for us because we could integrate this, now the effect of spacing and integrating clustering into an existing molecular clutch model. In this model, basically, we have an actomyosin-driven let's say contractility here, and then there is, of course, an adjustment in turn, also from what I told you before, of forces at adhesion sites. And spacing is really the key to maintain and have a certain distribution of forces that allow then to kind of maintain the contacts. So you can imagine that if you have a larger spacings and force get less and impaired, you can still maintain the contacts on soft materials. And to prove that, one can look at the actin dynamics. So, and looking at the retrograde flow, and there is, in fact, when collapse occurs, there is an increased retrograde flow in the cells. And if we play around now and we take rigid substrates where cells should form adhesion and regulate their length, depending on the spacing. And now we take a larger spacing, so adhesion should be very small. If we kind of release the clutch or we increase further the clutch, we have a growth of the contacts or a shrinkage of the contacts if we play around with the contractility of the cells. So having identified that actually our spacing serves, or it's a useful tool to actually understand we call it with Peter the molecular ruler versus like a force ruler. And then here we have really clearly like true spacing regulation of forces and not really like regulation, as I told you, which molecules exactly come into play. Now, we did some follow-up study to also connect, these pure findings on receptor mechanics and some cell functions. And what I'm showing here is we took some stem cells, mesenchymal stem cells, mapped also here the localization of myosin 2A and treated them with blebistatin. And then you see that the intensity of myosin signal is end up mirroring the ligand spacing, and we have a decrease if we treat them with blebistatin. At the same time, when we have an increase in spacing and then we start making the substrate soft or stiff, we see a similar behavior also in this cell type for the regulation of forces and traction forces, even observing higher forces for larger spacing as shown here, this quantification, and you can see those in the traction force maps. Also in the previous study, we showed that this is really nicely going hand to hand with the YAP activation of YAP-TAZ signaling, so for mechanotransduction. But interestingly, for these stem cells, one could even think of what is the participation in programming them to, for example, become bone cells. As we know from previous studies, also from the Disher lab, stem cells like to have rigid substrates to differentiate into bone cells, in presence also, of course, of the right soluble factors. What we started looking at was that the calcium signaling, and we saw again that it mirrored what we have observed at the periphery with adhesion signaling, and then also here for the uptake and activation of some channels. And for a long term, actually, this is even like on several days on the surfaces, we look at the classical differentiation markers for bone cells. This is alkaline phosphatase staining and alizarin red. And we saw, again, point out on soft substrates and larger spacing, because the cells, let's say, in terms of adhesion, felt good. Also, this is translated not only in activation of mechanical signal, but also in terms of differentiation signal. And to put this into a more comprehensive model, what we concluded is that, through adhesion and establishment of a very large contact, even on soft materials for large spacing, there is an ongoing adhesion and differentiation, osteogenic differentiation through the pathway, YAPTAZ pathway. I have shown you all the details on how we really look at the different steps, but in the end, the two signals converge. So in say that's for the, just like to show you two slides on the molecular regulation and how at the end these forces are adjusted and to assign some values. What we did was to use our platforms to regulate the clustering of integrins, and instead of binding just an RGD on the patterns as what I showed you so far, we use these springs that I mentioned as one of our tools. So there is still our favorite ligand, the RGD, and we use the gold as a quencher. And as the sensors get activated and pulled by integrins, these are like extensible springs that are recalibrated, we do see a force signal. And one can quantify this single, let's say, receptor force generation over time by monitoring this fluorescent intensity over time and looking at the effect of spacing. And you see again that from the mechanical regulation, only for the smaller spacing forces, as clusters increase in size, the mean tension per receptor is increasing, and then it's maintained also only for the small spacing. So, and this is again all connected to close to circle to actin. So this is a time lapse here showing that's a polarized light. Here we have a reflection interference contrast microscopy, then a live act, and that's our force signal. Hard to see it in the whole cell, but if you look at the snapshots, you always see first the actin and then some force accumulation. And this goes on as, you know, everything grows and has to get stabilized. So this is, I would say, where we are sending so far, yeah, for the regulation of integrins, integrin bonds. Of course, there is really a lot of room to expand now and apply these tools to understand several cell functions. I showed you, for example, for the osteodifferentiation, but you may think of, you know, endless applications of these pattern substrates to program different cell functions. Still, one thing that, you know, to really understand also the cell surface receptor work is that we should not forget that the integrins are not really working alone, yeah, at the cell membrane. And I don't have time to mention today, but even depending on integrin types, we have a lot of different, let's say, contributions in the regulation of adhesion. But what I would like to show to close this first part is how at the end we started integrating also regulation of receptor binding and mechanics with activation of different classes of receptors. The best partner or the most known partners for integrins are growth factor receptors. As you know, both, let's say, their ligand types are present in the matrix. And there are different studies that show all possibilities that can take place. And this is nicely summarized in this review from Manuel and Matt here of a couple of years ago, where you know, there could be collaborative signaling that comes as a secondary or enhancing activation of growth factor when the ligands are recognized, or a direct activation when integrins get activated, they kind of as a neighbor activate also growth factor signaling. And the other thing that has been reported also for the different growth factor receptors is that they might even co-participate and create promiscuous clusters even within focal adhesions and activate different type of signals. And one of the most exciting, I have to say, because the EGF field is really nice and a lot has been done, but again, is you know, what I find interesting is more like the regulation of family of TGF beta receptors, and in particular receptor tyrosine kinases such as BMP receptors. The reason is that because they have really a distinct way to activate cells to become bone cells. but also they participate in adhesion. Now it's really like a growing body of evidence that through these signals, they activate cell motility. And depending on co-activation also of transcription, that is dependent on the SMAD, a complex activation, there is also a differentiation, regulation of cell differentiation. And integrins here are really very important partners. And what is still, I think, puzzling or needs to be, looked in careful details is which integrins at the end talk with BMP receptors, and it might be really a lot cell type dependent. Like in this study from Korean Albish Rizzo, they nicely showed that here alpha V beta 3 is the right partner for BMP receptors when they use either soft or stiff matrices, and I've seen also co-regulation of cell adhesion and spreading. This is like factor added in solution versus factor trapped in a matrix. So that what we've been trying also with other groups is really to understand how these growth factors in the end may switch their function in regulating adhesion or differentiation, and this might have to do with type of presentation in relation to adhesive receptors such as integrins. So to look at this adhesion and let's say relation to integrins, we then used our nano platforms again. In this case, you have to think that the gold particles now serve as the anchor point for the growth factors. So we mobilize them with a bifunctional linker that we validated before in other studies, doesn't perturb the signaling abilities of the growth factors, although the factor is really covalently mobilized. So you have to imagine that the receptor sees it, gets activated. We showed also there is some trafficking of receptors in absence of intracellular transport of the ligand. So it's really matrix bound and not released. And the question here is how these growth factors function for the regulation of early adhesion when cells are also seeing integrins. So now integrin ligands are placed between the particles on a special type of PEG with a click chemistry. And we could immobilize two different types of integrin ligands, one selective for an integrin subtype, alpha 5 beta 1, and one that favors more alpha V beta 3. The reason is that these two integrins have different function in the process of differentiation of cells into bone cells. And we thought that some might be more kind of the right partner for BMP depending on the stage of adhesion and growth of the cells. This is an overwhelming slide with a lot of pictures, so maybe it's good if you just focus on the right side. And one thing that we did already in the past with these different type of ligands is that actually spreading heavily depends on which type of integrins you engage. So we have really larger cells in the presence of the alpha 5 beta 1 ligands. If we talk about fibroblasts, these are C2, C12 cells that are really highly responsive for BMP. What is really evident is that the co-presentation now from the surface with the growth factor and integrin alpha 5 beta 1 strongly enhances adhesion and spreading. Also for the RGD ligand, but I have to say the difference is still significant there from statistics. It's a little bit like really close, yeah, but here is really very evident. And if you add the factor in solution or it's absent, you really have the effect coming only from the surface. So soluble factor probably is triggering for this early adhesion time a different type of signal. And we look at focal adhesions now. What we really did, it's a complicated approach because it's kind of like you put one ligand on the surface and then you stain for the other integrin and see actually what is the co-recruitment to form more mature focal adhesions, because both integrin beta 1 and beta 3 are present in mature focal adhesions. And what we observed is actually that in this case, alpha-5 beta-1 is the right partner for the immobilized BMP2 to co-recruit beta-3 and make the contacts more stable, as is shown here and you see in these clusters. If you do the reverse and you give here just the RGD, everything looks pretty much similar. So as you know, the ligand is really dominating, not much of an effect in terms of focal adhesion growth triggered by the growth factor. And this is really recent work. Now we are trying to refine the system together with Carles, and Luis is a student in his lab, now to really create a one-to-one proximity of the two ligands with a chimeric engineered ligand where a single integrin and a single BMP receptor can bind on the same spot, and really see how with this one-to-one binding and recruitment, the crosstalk is regulated. What we could conclude so far in terms of adhesion, and of course this requires really then to do some traction force microscopy and really understand the regulation of overall cellular forces. What we know is that this co-presentation of the active mimicking BMP peptide with RGD here is good for adhesion, and all the rest is kind of like if you give either one, you have less adhesion, or if you block with integrin blocking peptide, you impair adhesion. There is also an effect on signaling with not really activation of the canonical BMP pathway, but some of the alternative ones. With this P38 is really, really elevated. We do not understand here what's going on. So if some of you have ideas really how to connect the P38, so it's like MAP kinase pathway to adhesion, this would be really, really great. But in terms of signaling now, to really close the loop and the last couple of minutes before we go to the cell-cell adhesion, is that, again, what I told you so far has been mainly focused on BMP signaling, regulation, or like putting some kind of connection at the periphery and seeing how this is regulating downstream growth factor signaling. Now, if we do it the other way and we say, well, we now start playing with cell mechanics and see how growth factors participate in the regulation of cell mechanics. One of the good candidates I mentioned to you before is this mechanotransduction pathway through YAP-TAZ, which is really like a nice switch. So on stiff materials, the pathway is on and activated, and it's off on soft materials. This is a summary slide of what we did in this respect, because from what I mentioned to you before, cells undergo through differentiation when osteogenic differentiation when they are on stiff materials and in presence of factors. But actually, if we put them on soft materials, so look here at the right side, there is still some triggering of signaling. What is ineffective is the YAP-TAZ, and that is really the crucial element for the co-activation of a transcriptional regulation for osteogenic differentiation. And we identify a lot of co-regulated genes that actually converge, and there is probably putative binding sites also of the YAP-TAZ to the SMAD complex to really make this transcription effective. And this is shown here, where in fact these binding sites have been identified. So it's really very likely that first the complex goes there, and then the YAP-TAZ is finalized in its activity in the nucleus. So what we did, it was really a funny way of perturbing cells, and it can take them on a soft material. So give the growth factors. We know there is activation of the signaling, but the signaling doesn't get effective all the way to the nucleus, as shown here. That's one of the transcription factors. It's called Osterix. It's all in the cytoplasm, and you see in the nucleus, it's like void. If you take YAP and now start overexpressing it, suddenly Osterix goes to the nucleus. So telling us that really playing around, with one or the balance between one or the other signaling pathway really is important in the regulation of this transcription of for osteogenic differentiation. And all this can be, even taken into a 3D environment with the soft materials. We do not have here in the 3D the level of control that I told you before with the patterning, where one can create kind of titrate the amount of growth factors that get trapped here in these hydrogels, and functionalization with integrin ligands. And then, for example, encapsulate cells, pre-program them inside these soft materials that in principle we know from what I showed you before will trigger the adhesion signaling and the BMP through the SMAD, up to the SMAD. And then by inserting a cleavable peptide, these hydrogels get, degraded over time, and the cell migrates to the bottom of the well or a stiff substrate. This is how the hydrogels look like. Right, you see there are more pretty ones around, but for us these are kind of sufficient to ensure that cells can even stay long enough for several days and get nutrients without suffering. And by varying this arm mixture with this different peptides, we can have again soft and stiff materials. And the gene expression is really showing what I was telling you before, is that in the end, the cells need to reach the final destination for a stiff substrate to differentiate. And this is relied on tissue culture plastic, BMP treated. This is the condition where they can start expressing osteogenic factors. In other cases, just on rigid substrate, no growth factor, or on soft material with the growth factors, or in absence, there is really very little or absent osteogenic differentiation. And for several days after, you see also differentiation as shown here with alkaline phosphatase staining. So here I summarize, and these are my key points to take with you. And if you have thoughts or ideas how to use this toolbox, please feel free to contact me. I'm very happy always to expand it and share with you our materials and reagents. So one thing is that this immobilization and different spacing, in the end, we control clustering and resulting forces. And we can look at mechanotransduction and the co-regulation of different signaling, look at the crosstalk. We still do not have really all the pieces here with localization of receptors. We've done also studies I haven't shown here, super resolution microscopy, to look which BMP receptor subunits will colocalize within focal adhesions, and how to analyze them also in cells. We developed some codes for receptor distribution. And in the end, I think that really the major advancement that brought us to this point is to couple this nano-pattern substrate to soft materials and look really at this combination of mechanochemical sensing. So now this is really like, I would say, the major part. And then for the second part, I will touch recent work that we did on the regulation of cell-cell adhesion, and how in the end we can also have a local switch and control cell-cell adhesion formation on also dynamics, and how in turn the modulation of these events has an impact on cell mechanics. So for local switches, some of you might already immediately think about optogenetics, right? It's like one of the most popular tools. And in fact, I mean, the light as a trigger is really, I would say, a very good system, because you can play around if you have good microscopes, and you can have subcellular resolution, really activate parts of the cell, be very short if you have really good systems, so with sub-second, I would say in quotes, is non-toxic, non-invasive. Of course, it depends on how you use light. And it's flexible in the end, because you can use with light microscopy, of course, equipped with right filters and the choice of labels that you have. And in our case, really, we made efforts to make it compatible with setup to measure forces. So the wishful thinking here when we started this project was like, oh, wow, we want to create a switch now to make a cell going from a collective here. There is, I don't know, some switch that we can create, and then the cell will start escaping and going. Yeah, it's not so simple, but somehow we got a little bit into that direction. But yeah, just to know, yeah, I mentioned the optogenetics tool. There is also like caging of ligands, which really requires a lot of knowledge in chemistry and a really perfect setup not to ruin your caged molecules when you don't want to, but they're really excellent tools, and some photo switching approaches. And the idea always like in this, for example, uncaging or light-induced dimerization, is that really you use in different cases a light, yeah, as I told before, as a trigger, and then you can favor some interaction of proteins. In this kind of, flow of thoughts, what we did, in fact, we took advantage of a system that already existed, and it's based on like the use of some small permanent molecules. They're chemical dimerizer, and some can be designed to have a photocleavable group here. And if you make them like be functional again, so with a ligand, let's say for a tag that you attach a protein of interest 1 or 2, in this case halo or a snap tag, when the cell is expressing these two proteins with the tags and you insert your dimerizer, it will go in the cell and start building or bringing the two proteins together through this specific interaction. And then the photocleavable group is actually the thing that will break when you shine some light. So conceptually, it's actually the opposite of what you do with optogenetics. You switch on something with the light. In this case, you break or disassemble something with the light. And these dimerizers at adhesion sites, I will show you the example for cell-cell adhesion. We're working on the TALEN also to build now focal adhesions. Basically, what you could do is the classical way that has been shown so far with the sequestration of some complexes to the mitochondria and deprive the cell for certain proteins. Or as in what I described to you before, recruitment of some, for example, partners, in this case, intracellular proteins that come along with active binding sites, and then you break them with light. This is what we've done with the alpha-catenin. Or you build even like a receptor. You bring together the cytosolic tail attached to this a part of the receptor, and you assemble again as a Lego blocks also here, the complex. So in this piece of work we did together with Richard Wurmbacher, a colleague from at that time I was at the University of Heidelberg. Now it's one of my neighbors here at the MPI, just across the corridor. Is really look at the playing around with these tools to control, first of all, the assembly of adherence junction with the dimerizer, apply light pulse, and observe how the dissociation dynamics of this complex will take place and how it will impact cell-cell adhesion and the generation of cellular forces. And we also used this Lego tool for the cytosolic tail of icadherin. So this is like more the alpha-catenin linkage, and this is within the icadherin. And this is how it looks like. So one tag, and this is like for the alpha-catenin, is labeled also with a cherry in red, and in green here on the right side is the icadherin with the GFP. And you see here over time that with the minus means like, the hours that it takes for the dimerizer to enter the cells. And then at the 0, there is an application of a short light pulse. And then you see how the contacts disassemble. And so immediately it's kind of fast to remove alpha-catenin, and the icadherin, it's a slow process And there are a lot of tethers that still maintain the cells. And when we look at the non-photocleavable, one can also really use the tool just for inducing formation of adherence junction in cells, and normally you would do that by inserting this. We have done it actually also in fibroblasts just for fun to make them look like epithelial cells. And what I told you before about the beauty of using light is that you can, in fact, apply to a full monolayer of cells or a subregion and really make, for example, the monolayer, you see the scatter light that you have in phase contrast when contacts are not really formed, then it's decreasing as the monolayer gets more and more compact and adherence junctions are formed. And now coming to forces, what one can do is the classical wound healing. We do a PIV analysis and see how coordinated the movement is, if adherence junctions are present or not. So without the dimerizer, everyone goes by himself. Yeah, it's a little bit chaotic. In addition, with the dimerizer, we have really this nice front and coordination where leader cells really guide the collective. And so here we show that there is an increase in the correlation length for the advancing monolayer, and that also the traction forces are regulated accordingly. So there is a building of cellular tractions at the front, and as we release adherence junction here, there is a drop in cellular tractions here. So this is really a tool that can be applied there for all different studies in cell mechanics. And in fact, we share it with other labs that have interest in looking at monolayer dynamics or rupture. So we hope that more will come soon in this respect. And one thing that we did actually with the lab of Roberto Mayor and together with Adam Schellert, postdoc in his lab, is to even apply this dimerizer and the tools in vivo, and looking how really the perturbation, yeah, even in a vivo setup can take place. And surprisingly, it worked really well with this different tag. I have to say we had to change some because not everything is super compatible, for example, with the SNAP. But with this kind of dimerizer that is more kind of permeant for the cells, we could get really nice induction of the formation of adhesion. So going like if you think the control is here, and then the assembly of these contacts, and then disruption after exposure with light. So there is one thing that I haven't told you, because not because I wanted to cheat, but it's like unfortunately a limitation of this system is that once you apply the light and you break everything, it's not really reversible. You would need to add dimerizers gain weight. That's the old ones that cleaved are depleted and re-establish the system. And it's actually what is nice with optogenetic, that you can switch on and off. And it's still like I have to say limitation of this dimerizer approach. Richard is working on some dimerizer that can be reactivated. We're not yet at the point here to introduce them for the adhesion sites. And what we thought about is actually an alternative approach to play around with cell-cell adhesion, and you can think of possible expansion also to cell matrix adhesion, of course, is to base this on DNA nanotechnology. So this is thanks to a new lab neighbor, also Kerstin Godfrey, that she started a lab a couple of years ago. And we started talking about, she's doing amazing work with DNA origami, how we can implement this in cells. And people have used actually DNA linkers in cells, but mainly like with a cholesterol tag and DNA strands. And what we're missing is still, all what I told you, this cytoplasmic linkage to the cytoskeleton and the actin. So to establish really a good kind of loyal, mechanically loyal linkage to the cytoplasmic part of cells, we designed a hybrid protein DNA system. where we maintain, in this case, all the intracellular part, and we just replace the extracellular domains of ecadherin with DNA linkers. And by using complementary strands, we make then the cell form the connections. We can play around with the type of strands to modulate the adhesion strength. And this is shown here, that when you use only one linker strand, it's not sufficient to really form the tight contact, and the recruitment of the cadherin is actually taking place only when we have a complete DNA linker. And this is also like snapshots and observation, also with the recruitment of actin. This tool is reversible because now if you put some invader strands that will kick out the complementary ones, it can actually disassemble the whole junction and then reform it again. So, and this is, I think, what makes life easier. Yeah, once you are through with the design and the expression of your hybrid system works. And it's shown here. We are, for example, with after even short, it's ultra fast system. I think compared to the hours that we had to get the dimerizer, still it takes a couple of minutes. But then we can disassemble our linked adherence junctions. And again, these complexes, yeah, it takes time to introduce them, of course, and also to validate that they're functional. And this is one of the proofs that we have a co-recruitment also of the catenins there as the junctions are formed. And there is some, we call it downstream signaling, I would say downstream recruitment here of the whole complex. As well as orientation, what I mentioned to you, of the actin fibers, to really say that we go from this transient or initial contact to a more mature with a bundle of actin. Questions is that in the end we show the molecular components are there right in place, and how can we really measure the binding strength and tune it. And for cell-cell adhesion, still I think it's not easy setup, but it's in the end of the day very rewarding system to use AFM and trap one cell on a cantilever here, and have a cell, the bottom cell that we want to probe adhesion for, on the dish. And these experiments have been done really in different setup. What we did here is really to use our linker and then measure first at the detachment, look at the detachment forces, look at rupture events, and see how this over time we can get eventually cell-cell separation if needed. And this requires really a lot of tricks. Also for students, it's just to tell you that some system can get very, very complicated to get at the end of the final goal. But eventually, with a lot of resilience, one can make it. And in this case, really the joint effort of Christina from the lab and Andy, who did his master's thesis between my lab and Kirsten's lab, was really the best combination we could have here in getting, the first step again, playing a bit with adhesion, making some Petri dishes that are compatible with the measurements, have some cells that are attached here, but from the same population, take some non-adherents to catch them, and then treat them with separately with the different strands, and then do the probing. So this kind of orthogonal strategy, we call it. And the experiment really looks like this, that you have cells and then you capture one and you go on top of it and you look at detachment forces as you increase the time. So let them touch 2 seconds, 5 seconds, 10 seconds. And sure, it's intuitive that as you increase the contact time, you increase also the force that is needed to separate them. And what we observed actually is that in presence of our linker, we exceeded even the maximum strength that we could record with a reconstituted full ecadherin. So we have this, and we use also different type of strengths, like we call it weak, intermediate, and strong linker, where I hope the movie is playing. No, sorry, it doesn't want to play. I'm really sorry for this. There is some issue here. But we do see also level of coordination, and this correlation length that I mentioned to you before is increased in presence of the linker. And you really need to have, to reconstitute the integrin here for the correlation of a stronger linker. So we still do not understand 100% why the stronger linker is stronger in terms of cell-cell adhesion and for coordination is performing as good as the control. For the velocity, it's actually a little bit more like efficient. So this is something to look in the future here. And with that, this is really like the summary here, what I told you today, that if we make use of different tools, that at the end one can get very creative for some of them. It is, I would say, nice strategy to, in the end, resolve some of the adhesive or the local regulation of adhesive interaction, the cell matrix and cell-cell adhesion. It is possible also to relate all these force events to other type of signaling, from what I showed you, for example, for the osteodifferentiation. And these tools so far we can apply at different length scales, some, all of them, of course, in vitro, and some in vivo setups. I would say there is a lot of room for expanding all these toolboxes. So with that, I'd like to thank the lab. This is still a picture in Corona mode. We should take one now. The days are more stable. The weather is nice again in Heidelberg. And I highlight here the main lab members, some current former lab members whose work I showed today, other lab members here. Lots of collaborations for different bits and pieces. And I have to say that's one of the winning things in doing interdisciplinary work, to work with people from different fields and really join interests. Okay, I'd like also to thank the funding for supporting our lab over the years. And I thank you for your attention and happy to take questions if you have any.
Very many thanks, Ada, for this fantastic talk. Yeah, anyone has questions? Otherwise, I'll start. But okay, so I'll start then. So Ada, in your sort of optogenetic tools, really, for the cell-cell adhesion, could you envisage doing the same sort of kind of approach, but just to bind it to the surface and not with the molecular tensors or DNA, but some sort of strategy that you can modify, and of some sort of integrins or GDs or something that you could sort of build up something that is approach with light? Is that something possible, you think?
So we tried that, and I think we failed because we're not really, I mean, we're not a chemistry lab here, I have to say, for the design of all these things. Yeah, we rely a lot on, and I know there are many groups that do nice caged peptides, and you can control that. So one is like from Arantxa del Campo, she did really beautiful work with the selective ligand. With dimerizer, actually, I think you asked because the original idea for designing, for example, this extracellular domain truncated cadherin was to use the dimerizer extracellularly.
Okay.
We didn't manage really to get it to work. We wanted to, link. That's why we had the snap tag, and then we thought, oh, if we do a similar approach, it didn't work in that case. So at the end, I mean, the DNA proved to be much easier. You just order the strands, they get there, and they attach very well. Yeah. And one limitation, though, with some of these tools, with the light, is due to the fact that we use gold particles. So, for example, doing super resolution microscopy with gold nanopatterns, this STED microscopy is not really coming out right because of the gold.
Oh, I see.
So I would expect also one has to really think about what kind of setup to.
Yeah, good. Any questions? No, let's see. Maybe just getting shy at the end of the series.
Yeah.
So if you see, Anna, if you just try to think about sort of like there's all these ideas, and you have shown that as well, and Shiva Shankar has also shown it, right? I mean, depending on the sort of design, if you have a triangle, or if you have like if you confine the cells in different shapes and ways, is there any way to compensate shape by putting the right ligands in a particular place? In a sense, can you trick, for example, the effect of the shape by putting the ligands far apart or closer together? Is that something that makes sense?
It makes sense. We haven't tried, so it's something actually to think about. That's really an interesting thought. The other way around, what we did with the selective ligand, we saw that spreading is dramatically increased on alpha-5 beta-1 ligands. So the cell looks really, let's say, simply speaking, very hydrophilic, as it almost has no tension, yeah. And on the alpha-V beta-3, they look more like what we are used to see when we look at spread in fibroblasts. So actually, it's this interesting thought, because we haven't tried to do that, to couple the selective ligands to the shape now with micro-nano patterning, for example, and look at the different also generation of forces here. Yeah, so it's something.
And
yeah.
And the other question is, just following up. So obviously different cells react. I mean, this is a very fascinating field of integrins, right? For which I don't know much of it, but essentially they're very selective in which. So can you somehow mix and match integrins, in the sense just trying to, if you couple one integrin which you know is going to have full effect, if that makes sense, and the other one which is going to have half of the effect, and kind of again, can you sort of trigger binding by how to speak, sort of again fooling the cell by just putting different integrins at different levels and just trying to compensate one with the other one? Is that something that makes any sense?
Yeah. So we did that in stripes some years ago to see the segregation of, again, alpha V beta 3, alpha 5 beta 1 integrins in a fibroblast. But it was just for the sake of making the material
Right.
and showing that it works. How mechanics will be translated, I guess that you will clearly see different force contribution, and I wouldn't be surprised if actually for the beta one you see higher forces, because in the end you have what I mentioned to you, the secondary recruitment of alpha V beta 3, while the beta 3, alpha V beta 3, when it has a lot of RGD, it's kind of kicking out the alpha 5 beta 1. So this makes totally sense to me too, and it's probably in the end what is really taking place at focal adhesions, that you need to have both, and then the translocation of the alpha 5 beta 1 later as adhesions mature.
Good. So I can see some hands. So Carlos.
Yeah. Okay. Carlos.
Hello, Ada. Yeah.
Hi, good to
Good
see
to
you.
see you. Yeah, greetings from Barcelona. So thank you for the very nice talk. It was a fantastic overview of your recent work. I enjoyed it a lot. So I have a question a bit related with integrins. So I would like to know, based on your experience and your recent work, which do you think is the ideal partner for BMP receptors to have this synergistic signaling? Because I remember with UIS, we did some studies and it seemed that blocking alpha V beta 3 had a more detrimental effect to block the synergy than blocking alpha 5 beta 1.
Yeah.
But from what I understood from your talk, it would seem that alpha 5 beta 1 has a more important role. So I'm a bit confused. I would like to know your opinion.
I think it's two things, because for the early addition is alpha 5 beta 1, but then alpha V beta 3 has to kick on. If you block it, and it's seen also with the silengitide experiment, even with the Deviva, sure, you need to have the beta 3. So what I meant with what we found out is that really for the initial spreading,
okay,
and for the growth of adhesion, that's really, you need to have some beta 1 proximity, or it's more favorable. Yeah.
So do you think then it would make sense, now I'm thinking in further studies, maybe not focusing too much on the specificity of the integrin, but having an RGD ligand that is quite, I mean, not selective for one or the other, but that it's active for both integrins. Would that be a better approach?
Yes, I think so.
Okay.
So we should think about that to have more like a wide type, yeah, and maybe pay attention also to the stage of the cell. You know? So C2C12 is one story, mesenchymal stem cell is another story. To really say, okay, what stage also of adhesion we want to look at, or long-term effect. Yeah, I mean, for everyone else, I think the experiments we're doing is also in low serum to avoid co-participation of other factors. This we have under control in our system. But I agree with you that at the end, to have a more generic will kind of, you know, give or start the system, yeah, as we want. We won't know, yeah, in the end, which integrin is doing what at a specific time. So,
okay.
For that, we have the selective ligands, right?
Okay, good. Yeah. So, yeah, once again, thank you very much.
Yeah, thank you, brothers.
Thank you. Eileen?
Hi, Eileen. Hi, good to see you.
A really nice talk. I just had a sort of a specific question going back to the polyacrylamide and your controlled systems on there. And a few years ago, there were papers that were suggesting, oh, there's issues with binding on polyacrylamide because the porosity of the material. And then other papers said, no, that's not really a thing. And I know it may not matter with exactly how you're doing your systems, but did you have any thoughts on that? Like, about whether we can just trust polyacrylamide as a system to modulate stiffness?
So the short answer is we can't trust it. You know, and that's why there are many other type of soft materials. Yeah, we're trying to use an alternative. Yeah. It's of course, I mean, our control is like in the absence of ligand, so we know a little bit, let's say, more control because of the particles in there. But I'm with you, yeah, and I mean, we have also done the transfer of particles on PEG, for example. Some of the results are really similar. Now we started using these elastomers also, and there you see, okay, there is also the viscoelastic component that is totally different than what we have with the polyacrylamide. So I agree with you that it's not really the perfect material, and whatever is going on at the surface is.
Okay, yeah. But you would have more faith in PEGs to do it.
Yes, exactly. Okay. Yeah. Which is, I don't know if you share the same thinking with it.
I do. We have data we haven't published, but I have other reasons to doubt it too. But thank you. Thank you.
Thank you.
Good. Thank you very much. Is there any other last minute question or otherwise? Nope. Okay, so in that case, we'll thank Ada once again for this fantastic talk and for closing our series of seminars this semester. Thank you, Ada, and hope to be in touch, hopefully.
Yeah.
All right.
Thank you very much.
Thank you very much.